So I left off, we were talking about the way that we name species and the rules of naming, which were developed in the 1700s by a guy with an absolutely monster ego who was on a personal mission from God to understand God's creation, a chap by the name of Carl Linnaeus. And he came up with the convention of using binomial names for species, which means that every species is given a formal name in two parts. The first refers to the larger grouping that it's a part of, that's the genus. And the second part refers to which precise member of the group it is, which is the species name. Not that I care that much, but just so you know, formal species names are capitalized, in Latin, they're in italics. We capitalize the genus name, but we never capitalize the species name. So the bluegill, one of the tastier fish in Arkansas waters, is here called Lepomus macrochirus, which the transcript called Lipomas macrocongrus, whatever. And Latin's a very flexible language, and lots of words have been borrowed into it. And technically, Latin names don't have to be the Latin that the Romans would have recognized. They can be derived from other languages. They can even be made up. There are some that don't mean anything before the scientist just decided that that was what he was going to use. That just came up with, names like Bobana, Cocana, Dodana, or some such. But they still have to be spelled in the Latin alphabet. They have to be pronounceable in Latin, and they have to follow some boring rules of Latin grammar that we don't need to get into. There's a bunch of other rules that govern what happens if two species accidentally get the same name, which, given that we have 1.8 million species to keep track of, happens more often than you think. If two species get the same name, one of them has to be renamed, and there are rules of priority for deciding which one gets what. So that's a species that doesn't have a name, or at least not a genus name, derived from Latin. Ginkgo is a Latinization of the Chinese name for this tree, which we have growing around here, including a couple of nice ones in the back of the Lewis Science Center. Many of you have seen ginkgos before. And then Abu Defduf is a rather pretty tropical fish from the Red Sea. And Abu Defduf is not Latin, it's the local Arabic name for this fish. So names can be borrowed from any language that you can think of, and have been borrowed from a great many. They just have to be writable in the Latin alphabet and follow some grammatical rules that we don't need to get into. I'll note that the describer of a new species gets the right to name it. And the describers, you're not really supposed to name a new species after yourself, but describers will often name species in honor of others. And a lot of very old plant names that have been adopted into English were originally coined to alter fellow scientists. So this guy's name is Georg Kamel. He was a missionary in Asia in the late 1600s and early 1700s. He was one of the first Westerners to describe the plants of Asia to a Western audience. And Carl Linnaeus honored him by naming this beautiful Asian flower Camellia, which is derived from the Latin version of Camel's name, Camelus. So camellias and gardenias and forsythias, that's that yellow flowered plant that's one of the earliest around here to bloom in spring. Wisteria. There's a bunch of common plant names that are ultimately genus or species names that were named in honor of somebody. And the tradition continues to this day. On the left, that's a moth that was named in 2017, Neopalpa Donald Trumpi. It's native to the US-Mexico border, and it has this covering of golden scales on its head. You're looking at its head from the side. So let me get out the pen. There's the eyes. These are the mouth parts. And on top of the head, you have this big covering of golden hair-looking scales on top of its head. And it is fixated on the US-Mexico border. And I'm told it has unusually small genitalia, which I'm sure has nothing to do with the fact that it was named Neopalpa Donald Trumpi. This is a fly native to Australia that was named in 2011. And you might notice that it's got a posterior that is this beautiful golden yellow color. And it was from the loveliness of its posterior that the scientists named it Scaptia beyonceii. Yes, it was named after Beyonce. There's a number of rather sly little jokes that work their way into scientific nomenclature. There was even a biologist in Germany in the 1930s who named an insect after Hitler. And it's still around. And it's whether this was an honor or not, I'm not entirely sure, because the insect is completely blind and lurks in caves. So it's not like they named a new species of eagle after Hitler. If you've got to name something after Hitler, a tiny insect that can't see and lives in caves is probably a pretty appropriate choice. Okay, anyway. Oh, there was actually a fossil named after me years ago, and it's a weird old fossil and nobody is quite sure what it is, which is perfect. So you could look up Yorgia wagoneri sometime if you really, really wanted to. Anyway, names don't have to be accurate descriptions. This happens to be a bird that spends so much time in the air that it was once believed that it didn't have any feet. Nobody ever saw it landing. It actually does have feet, but its name, Apus apus, is Latinized Greek for no foot, or since it's twice, no foot, Apus apus. And that's just an example of the principle that names do not necessarily have to be accurate descriptions. They're just names. You know, just like the fact that my name is Wagoner does not mean that I still drive wagons, although presumably I had distant ancestors that did. Let's look a little more closely as to what we mean by species. I've been kind of vague about it, but let's get a little more rigorous here. Linnaeus defined species on the basis of distinctive physical features that they have. Here's another tasty and fun fish in the freshwaters of Arkansas and other states. It's in the genus Micropterus, Gesundheit. And right there's the eye, of course. And this thing right here is the upper jawbone. And I'm outlining with the pen. And you can see that the back edge of the upper jawbone extends back beyond a vertical line that's drawn so that it just grazes the posterior side of the eyeball. See what I mean? I'm drawing this vertical line that just touches the eyeball, and the upper jaw extends behind that. That makes this Micropterus salmoides, the largemouth bass. If it had a smaller mouth and the upper jaw did not extend past the rear edge of the eye, we might have Micropterus dolomieu, the smallmouth bass, or Micropterus punctulatus, the spotted bass. And so species are defined by distinctive and sometimes very subtle physical features that they've got. Sometimes you might have to count scales on a particular body part to tell what species of fish you've got. Or with insects, you often have to look at very fine details of the genitals to identify to species. It can be pretty obscure features. The problem you run into is that species tend to be variable. If you think about this class, we're all members of the species Homo sapiens, and yet we vary a lot in height, in build, in skin color, in hair texture, in eye color. We vary in many thousands of ways. Here's another very variable species. This is a Cuban land snail. They're all classified in one species called Polymyta picta. And you might think we ought to divide this up into separate species because there's lots of different types of snail right here, but it's not obvious where you would draw the lines. You might put, let's say, all of the brown snails into one species, but the brown kind of overlaps with the yellow. There are some of these that are kind of yellow-brown, and maybe it's hard to tell which color they should really be. The brown tends to overlap with the black. There are some that maybe are closer to black than brown, but where do you draw the line? It's not obvious looking at all of this exactly how you could divide this set of snails up into separate species. So members of populations of living things vary amongst themselves. Sometimes the variation is not obvious unless you really look for it. Sometimes it stares you right in the face. So how do you decide how to divide up living things on the basis of what they look like? Well, the answer is these days we generally try not to, because it can be very subjective. Linnaeus used what we now call the morphological species concept, and that root morph means shape, means what something looks like. And in Linnaeus's morphological species concept, a species was defined by physical features. And as we've seen, a lot of species can vary their physical features quite a bit, and this can be ambiguous. This makes the morphological species concept ambiguous to use in practice. There are a number of alternate species concepts that have been proposed. We don't have time to get into all of them. This is a pretty big field. This is a lecture's worth of material in one of my upper division courses. But for our purposes here, you need to know about the biological species concept, which defines a species by its members' potential to interbreed. Members of a species are potentially able to interbreed and produce fertile, healthy offspring with each other. Every human on the UCA campus, in principle, could interbreed with every other human. I mean, some people might have, medical conditions or things like that, and I'm not really suggesting everybody should try it. But in principle, we could all interbreed with each other and produce fertile offspring. There's no group of humans that can have children amongst themselves, but can't have children with any other humans. And we call that reproductive isolation. A species is defined as being reproductively isolated from members of other species. If you could mute your mic, whoever that is, please. Members of... Could you mute your mic, whoever I'm hearing? Okay, thanks. Members of one species usually cannot produce fertile offspring by mating with members of other species. Very simple example, horses and donkeys. You can get them to breed, and they do produce offspring called mules, but those offspring are not fertile. The offspring cannot breed themselves. Mules are sterile. And for that reason, we call horses and donkeys separate species. And we sometimes find species that you can't tell apart by looking. This is a species you can find around here, especially in winter, especially if you set out bird seed on the ground. On the left, that's the Carolina chickadee, Pusile carolinensis, and that's the bird we get around here. On the right, north of about St. Louis, a different species of chickadee lives. It's called the black-capped chickadee, Pusile atricapillus. And as you can tell from comparing the photos, they are practically impossible to tell apart just by looking. We call these separate species because they don't interbreed, because they never get together and lay eggs and make babies with each other. Part of the reason for that is that they've got different songs, and birds often attract mates with very specific songs. Females of the Carolina chickadee don't respond to the songs of the black-capped chickadee, so they never have any babies. In cases like this, we would call these cryptic species. We call them separate species by the biological species concept. By the morphological species concept, we can't tell them apart. But even though the morphological species concept is sometimes the most practical for everyday use, we try to get away from it and stick with the biological species concept. There are cases where that species concept can be ambiguous as well. If you really want to know the details, I talk a lot about it in Biology 4415, but this is 1400, so let's keep going. Onward. Now, remember that species that are similar go into the same genus. So these are 3 species out of 12 or 13 in the genus Lepomus. Lepomus macrochirus is the bluegill. Up there at the top, Lepomus gibbosus is the pumpkin seed. And Lepomus cyanellus over there is the green sunfish. And there are more. Again, I think there's 13 North American sunfish in the genus Lepomus. I don't have space to show them all. All 3 of these live in Arkansas, and they're the kinds of things that you could catch. Although I think the pumpkin seed's usually too small for eating, but we do have it here. Genera that are similar, this is the other big thing that Linnaeus did. He came up with what we call a taxonomic hierarchy, where every unit at one level can be grouped with others into a new unit at a higher level. So species that are similar get put in the same genus. Genera, which by the way is the plural of genus, one of those Latin plurals that doesn't follow English rules, so it goes. Genera that are similar all get placed into one family. So at the lower left, we've got 4 of those 12 species of genus Lepomus. Up at the upper left, those are 2 species in the related genus Pomoxis, which we call crappies. These are the fish that my cross-the-street neighbor keeps taking his boat down to Lake Conway to try to catch, and apparently usually doesn't. And then at the lower right, those are 2 species out of maybe 6, I think, in the genus Micropterus, the freshwater black basses. So this includes largemouth bass, smallmouth bass, not striped bass is different, spotted bass, Alabama bass, maybe a couple of others go in genus Micropterus. The striped bass that they catch down at Greer's Ferry are actually in a separate genus. All of these genera go into one family called the family Centrarchidae. And then a bunch of similar families go into a larger group called an order. In this case, it's the order Perciformes. And then one or more orders get grouped together into a larger grouping called a class. And then one or more classes get grouped into an even bigger grouping called a phylum. And then multiple phyla get grouped into an even bigger grouping called a kingdom. And several kingdoms get grouped in an even bigger super duper grouping called a domain. And it looks like this. I'll take it in the opposite direction. Our bluegill belongs to the domain Eukarya. That's all living things whose cells contain little lumps of DNA called nuclei. I'll talk about that in a bit. So everything from people to sponges to microscopic paramecium to mushrooms to oak trees to fish goes in the domain Eukarya. We divide that domain Eukarya into multiple phyla, multiple kingdoms, and the fish goes in the kingdom Animalia. That is organisms that have nuclei in their cells, and they're made up of many cells, and they go through development as embryos. And then the kingdom Animalia is divided into roughly 30 phyla. Phyla is the plural of phylum. And our fish goes in the phylum Chordata, which is all animals that have a nerve cord running down the backside, a spinal cord. And then the chordata can be divided up into multiple classes. And we put the bluegills in the class Actinopterygii. That's chordates that have fins that are supported with flexible or bony supports called fin rays. You can see the rays really well. I'm drawing them in right here. They're these spiky things that are holding up the fin. And some of them, like the ones in the tail, are a lot more flexible. That's the fin rays, and all chordates that have fin rays go in the Actinopterygii. That's divided into many orders, one of which is the order Perciformes, the fish that look like perch and have sharp rays at the front of fins and flexible ones at the back. Perciformes gets divided into many families, one of which is the family Centrarchidae. The Centrarchidae contains many genera, one of which is genus Lepomus. And genus Lepomus contains 12 or 13 species, one of which is Lepomus macrochirus, the bluegill. The closest analogy I can think of is something I always get wrong, but those of you that have a military background can probably fill me in on this better than I can. If you look at the organization of the army, right, you start with the individual soldier. You group several soldiers together into, I think, a fire team.
A platoon.
A platoon. Okay, and then you group several, I thought you grouped several teams into a platoon.
Well, you have several teams. So usually you have about 13 fire teams, and that creates a platoon, at least from the Marine Corps point of view.
Okay.
Yeah, there's no fire teams or whatever in the Army.
Yes, you're
Okay.
talking about squads, because they come in squads as well.
Yeah, that too.
Okay,
Yeah.
all right. This is why I get confused. It's partly because different services have slightly different rules. but multiple soldiers get grouped together in a platoon. Multiple platoons get grouped together in a, help me out,
Company.
in a company, that's right. Multiple companies will get put together in, is it a regiment?
I mean,
Battalion.
Usually a
yeah,
battalion,
battalion.
In a
and then the
battalion?
regiment is over the entire battalion,
Okay, so
or
multiple,
over all the battalions, really. But yeah,
Or a brigade.
okay, multiple companies get grouped in a battalion, multiple battalions in a regiment, and so on. I'm going to stop there because I always get the details wrong, and it's different for different branches of the service. But the idea is the same, where you can group bodies of soldiers at one level. You can take several of these groupings at one level and put them into a larger grouping at a higher level. So multiple soldiers in a platoon, multiple platoons in one company, multiple companies in a battalion, and so on, all the way up to, the 5, 6 now divisions of the U.S. Armed Forces: Army, Navy, Air Force, Marines, Coast Guard, and we've got the Space Force now. Okay, that's the closest analogy I can come up with to the way that biology groups species. They group them in this hierarchical grouping in which each grouping has a rank. And the ranks are domain, kingdom, phylum, class, order, family, genus, species, which you can remember with the sentence, did King Paul come over from Germany swiftly? Or do kings play chess on fat green stools? Or do kinky perverts come out for good sex? Or whatever you want to do to remember that. And I won't go through that for all of life, but I'll start at the very top with the 3 basic domains of life. We can classify every living thing that we know into 3 huge groupings called eukarya, or alternately eukaryotes, archaea, and bacteria. Eukaryotes are made of cells, and the cells contain flexible sacs in which the DNA is stored, and those sacs are called nuclei. We will talk a lot more about DNA and exactly what it does later on. Let's not jump ahead of ourselves just yet. And eukaryote cells don't contain just nuclei. They're very compartmentalized, and you have different, quote, compartments, different substructures inside a eukaryote cell, each of which does a different thing. It's loosely analogous to the way that your body is organized. You're not the same type of cell all the way through, but your cells are grouped into different organs that are specialized for different functions that keep you alive. So just as you contain organs, each of which does something different, eukaryotic cells contain substructures that do different things, and we call those organelles. So a eukaryote is made up of cells that contain nuclei and various organelles. And towards the end of this course, you'll get a guided tour as to what some of those are. We don't need to get into that just yet. The other 2 domains don't contain nuclei. They do have DNA, but the DNA is not packaged into a sac. The DNA just floats freely inside the cell. Cells that are organized like this are called prokaryotes. There, I'm boxing in the word. So eukaryotes, the cells have nuclei. Prokaryotes, the cells do not have nuclei. And we have 2 domains of prokaryotes, archaea and bacteria. Both of those, the cells don't have nuclei and they're not partitioned off into organelles. The differences between archaea and bacteria are pretty major, but they are differences that involve getting fairly deeply into biochemistry. It's not something that you're incapable of understanding. You could understand it perfectly well given the time, but biology is a huge field and we've got an awful lot of ground to cover. And this is one of those points at which I'm just going to punt the ball and say, if you really want to know, go check Wikipedia or check your textbook if you've got it. I'm not going to go into what makes archaea and bacteria different from each other. Just take my word for it that they are very different when you look at the details of their chemistry. The only thing I'm really going to focus on with archaea is that many archaea, although not all, live in environments that are too hot or too cold or too acid or too alkaline or too salty or too full of heavy metals for anything else to live. Many archaeans may be the only living things that can survive in the habitats that they colonize, and organisms that can live in these extreme environments, these environments where even Bear Grylls would not survive, are called extremophiles. Phile, by the way, is the Greek root meaning love. So extremophiles love living in extreme environments. Whoa. In an upcoming lab, you are going to see some eukaryotic cells nuclei. These are cells from the inside of a human cheek, stained with a special stain called methylene blue. Normally, they're transparent. The stain makes them more visible. And these cells from the inside of the cheek, each of them has this little dark spot in the middle. That's the nucleus. That contains the cell's DNA. You're also going to look at cells of a water plant called Elodea. This is coming up in lab 2. And Elodea cells look a little like bricks. So I'm outlining one of them right there. And I promise you there is a nucleus in these guys, but it's transparent and you're going to have a hard time finding it without using some of those stains. So there is a nucleus there. I'm just kind of drawing, that's not necessarily where the nucleus is. I'm just drawing it in as a reminder. But what you can see are a whole bunch of these green dots that you'll see floating in the middle of the cell. And if you watch carefully, you can watch them moving through the cell. Those green dots are a type of organelles. They're called chloroplasts, if you're curious. And they're what carries out photosynthesis. This is where the plant harvests solar energy and uses solar energy to take carbon dioxide gas and water and assemble them into larger molecules like sugars and cellulose. So you can see nuclei in eukaryotes and you can see organelles. A lot of their complexity can't easily be seen with light microscopy because a lot of it is transparent and or smaller than light microscopes can see. There's a physical limit to how much you can magnify using light. So if you look at a slice through a eukaryotic cell using a different type of microscope, this is called a transmission electron microscope. The nucleus is here. This is what I'm outlining. And it's full of grainy stuff. That's actually DNA, and we'll talk about that later. That's the nucleus. Inside the nucleus, this thing I'm outlining right now, this dark spot is called the nucleolus. Well, we don't need to worry right now about what that does. And what I want you to get a feel for is that outside the nucleus, there's a whole bunch of complexity. You've got these dark shaped colored things and you've got these wide open blobs, and you've got these wide open blobs containing grains, and you've got grayish blobs like this. Don't worry about the details right now. What I just want you to see is that this is a cell divided up into a lot of separate areas and separate compartments. And we'll talk much later as to what all of these do. This is a partitioned up cell. If you compare that to this bacterium, the inside of the bacterium is basically just kind of grainy. There isn't any nucleus. There's some kind of darkish spots, but they're not really separated from the rest of the cell. The cell is not divided up into organelles. This is a prokaryotic cell, and both bacteria and archaea are organized like this. If you need an analogy, think about buildings on the UCA campus. The Lewis Science Center or the library or most of the academic buildings are divided up into multiple rooms, and the rooms might be specialized for different things. I know some of you are in the library right now, and you might note that there are specialized rooms for individual study, there's group study, there's librarians' offices, there's workshops where cataloging takes place. There's workshops where sorting and reshelving is done. There are special rooms for the heating and air conditioning. There's different rooms in the library building, and they're used for different things. A prokaryote would be a little bit more like the football indoor practice facility that used to be called the Pepsi Center until Coke outbid them for the naming rights. I don't think I've ever actually been in there, and I don't know if this is perfect because maybe there's a few little offices on the side or something, but the vast majority of that building is one great big room, a room the size of half a football field. And most of what goes on in the football practice facility takes place in that one great big room. That's more like what a prokaryote is like. Carolina, we're coming up on that. That's more like what a prokaryote is like, one big room where everything happens. And a eukaryote is more like an academic building with lots of different rooms where different functions take place. Okay. Now there's two types of prokaryotes. There's the ones in the domain Archaea, and there's the ones in the domain Bacteria. They're both prokaryotic, which means they don't have nuclei and organelles. They're both typically very small. We measure the size of microscopic things in units called microns. And a micron, or you'll see it written micrometer, is 1/1,000,000th of a meter. If you can't figure out just how big that is, a single human hair depends on the person, but it might be somewhere between about 20 and 50 microns thick. And so typical bacteria might be maybe 1/20th the size of the thickness of a human hair. That's how small they are. By the way, you might see this at some point. When we abbreviate micron, we abbreviate it like this. That character over at the left is the lowercase form of the Greek letter mu. Those of you in frats or sororities, I'm sure, know the Greek alphabet, and mu is the Greek equivalent of M, and the capital looks like our M. The lowercase is written like this, kind of like the letter U with a dangle off to the left. So if you see that symbol, that's what we're talking about. So archaea and bacteria, both very small, both karyotic. What makes them different from each other includes some rather complicated biochemistry that we don't have time for. So, Caroline, I'm not probably going to be able to satisfy your curiosity as much as you might like. This is something I'm just kind of going to have to gloss over so that we can finish the course in time. They are different enough that we put them in different domains, but I'm going to have to be a little coy as to why. What you most need to know is that many archaea live in environments that are so extreme in some fashion that almost nothing else can live there. This is an aerial photo over the south end of San Francisco Bay. The Cargill Salt Company owns this land, and what you're looking at is a bunch of shallow ponds. And what they'll do is open a gate and let these ponds fill with water from the bay, and then they close the gate and just let the water start evaporating. And the ones where the water hasn't evaporated too much are this kind of muddy brown and green color. But many of these ponds have turned bright red, and the squarish ponds over here are a definite purple in color, the ones over on this end. And then the white ones have completely dried up, and then the salt company, Cargill, bulldozes up the salt and packages it and does whatever with it. But these ponds over here look purple because they have become so salty that almost nothing can live in them. You drop a fish in there, the poor thing would die horribly within seconds. That's how salty that stuff is. But what's making it purple is the fact that an Archaean species can live perfectly well in extremely salty conditions. We call this a halophilic Archaean. Halophilic is Greek for loving salt, because it can tolerate salt conditions that nothing else can live in. So Archaeans can be extremely tough in that regard. This is Grand Prismatic Spring and Yellowstone National Park. Just for scale, over along here, you can see that's a boardwalk with people standing on it. These little specks right there, those are people. So this thing is a pretty good sized pond. And you'll notice that the water's deep blue in the center, but gets kind of greenish towards the edge and then has these fringes that are yellow and orange. Yellowstone is volcano territory, and the water coming out of Grand Prismatic Spring, I forget, it's something like 150 Fahrenheit, which works out to 60-ish, 60-something Celsius, hot enough that if you, oh, somebody's been there, and yeah, it's pretty awesome. But there was a case a few years ago where somebody's dog jumped in, and the guy jumped in to save his dog, and both of them died in a very nasty way. It's way too hot for humans to survive in it. You know, any fish that you put in that spring would be parboiled in seconds. But the colors result from extremophile Archaeans that can live there quite comfortably. And I just suddenly realized I am low on time. So real quick, there are extremophiles that can live in acid rivers like the Rio Tinto in Spain. And there are extremophiles that live in superheated, sulfur-rich water coming out of the deep ocean a mile below the surface. This stuff that looks like black smoke is actually superheated seawater. And there are Archaeans that can live here at temperatures over 220 Fahrenheit. So that's all I've got to say about Archaeans, and I'm going to stop the recording now so we can cuss when it's over.